Welcome everyone. In this lecture, we are going to look at the topic Genomic Large Language Models. So this will be the last lecture of module 7, that is probabilistic and machine learning sequence models. So we finished the topic of hidden Markov models in the last 2 lectures. We saw how we can use hidden Markov models to model DNA sequences. Now one question I want to address before we move forward is how do hidden Markov models compare to, let us say, neural network based deep learning architectures? Well, there are some positive as well as negative points. So I want to emphasize that hidden Markov models are still used in bioinformatics for many applications. There are 2 strong points. One is interpretability, that is HMMs are a clear probabilistic framework for it's a very clear probabilistic framework for modeling sequencing data, and it makes it very easy to interpret how the model is behaving and how is it making the decisions. Further, HMMs are also more efficient and more accurate when we have limited amount of learning data. But on the downside, the limitation is that HMMs have limited representational power, so they are not able to capture complex patterns and dependencies in the data. So in this lecture, we are going to look at one class of neural network based deep learning architecture for modeling DNA sequences. In particular, we will focus on this genomic language model, that is a large language model trained on DNA sequences. Most of the content that I will be using in this lecture will be drawn from this review article that was published in 2025. So this article talks about the genomic language models trained on DNA sequences. So it talks about the review, like what all work has been done on this topic, what are the opportunities, and what are the challenges that remain to be addressed. Now I want to emphasize that in this course particularly, we have not discussed a lot of prerequisites that are needed for understanding these language models in detail. For example, we have not discussed neural network architectures, or we have not discussed basic topics in natural language processing. So as a result, I am going to discuss this genomic language models at a very high level, assuming that the students who are not familiar with neural networks can also follow this lecture without any problem. So just to start, so I am sure all of you have seen and used language models in your day-to-day activities. For example, ChatGPT is one of the very popular LLMs that we can use for a variety of activities like summarizing content, question answering, problem solving, etc. Now, given this remarkable advance in natural language processing, the question is how can we extend these techniques to biological sequences as well. Now before we answer that question, let us understand a language model. What do we mean by language model at a very high level? So a language model assigns a probability to sequences of words or tokens. For example, if you see these 4 sentences in English text, then a language model would be capable of assigning different probabilities or likelihood to each of these sentences based on the observed training data it has seen during the training process. So from the training data and from the training process, it would naturally learn to assign the highest likelihood to this sentence over other sentences, because the other sentences do not follow a proper grammar, right. So in this case, a language model would have learned to assign the highest probability to the sentence which makes grammatically sense, right. So that's a high level description of what a language model is. That is, it is assigning probabilities to sequences of words or tokens. Now when we are training these language models, there are 2 different types of learning objectives that are commonly used. One is called as next token prediction, in which basically the training of the model involves learning the conditional probability distribution of the form probability of the tth token being Wt, given that W1, W2 and so on until Wt minus 1 are the previous t minus 1 tokens in the sequence. So, this is conditional on the previous t minus 1 tokens seen in the data, and we are assigning a probability of the tth token in the current sequence. So, for example, in this case, as an example shown below, the 3 available tokens are Hannah is a, and the missing token that the model is being trained on to predict is the 4th token. And in this case, the model would learn different probability values for different words in the vocabulary. For example, Hannah is a sister, Hannah is a friend, Hannah is a marketer, and so on, right? And depending on what patterns have been observed in the data, the probabilities of different tokens would vary, right? So, for example, the probability of the word brother instead of sister would be extremely low, because that pattern would be seen much less in the data. Now, this type of learning objective is called next token prediction. There is another commonly used learning objective which is called as masked token prediction. Here, a language model is trained to learn the conditional probability distribution of the form Wm, probability of Wm, where Wm is the masked token that is being predicted, given the surrounding tokens, that is W1 to Wm minus 1 and Wm plus 1 to Wt. So, here the masked token is not known to the model. So, in this case, for example, the example is shown below, where example could be that I mask the middle word between Jacob and reading, and in this case the model would be trained to predict the middle word. So, it would assign different probabilities to different words from the vocabulary. For example, Jacob fears reading, Jacob loves reading, Jacob enjoys reading. All of these words may get significant probability because they make grammatical sense and they would have been seen in the training data as well. So, in this case, this masked modeling as well as next token prediction, they both are commonly used learning objectives. The next token prediction that we saw in the previous slide is the one which is used in the GPT models as well, because GPT models can synthesize new text using this next token prediction techniques. Whereas in cases where synthesizing new text or new language is not needed, in those cases masked token prediction may be preferred. Okay, so these were the two learning objectives, like what a language model would be trained to learn the probability distribution form of. So, moving ahead, now given these, so these are very simple learning objectives, but one can ask, okay, what are the primary contributing factors which led to the recent breakthroughs in natural language processing? One is the use of complex models, that is state of the art deep learning architectures that fit the observed data. And second is that these models are trained on large amount of publicly available data. So, for example, the latest versions of GPT models are trained on trillions of tokens. So, given the vast amount of data and the state of the art neural architectures, they are able to generalize well to a large number of natural language processing applications. So, as an example, like the shown below is this comparison between GPT-2 and GPT-3 models. You can see that the GPT-3 is extremely bigger deep learning model compared to GPT-2. So GPT-3 has 176 billion parameters, making a very huge model, and it was trained on a large data corpus of 570 GB comprising books, articles, websites and more. Now the question is how best to adapt and extend these techniques for biological sequences, for example DNA, RNA, proteins, and this is a very active research topic. Now one reason for why researchers are interested in extending these techniques to biological sequences as well is because we also have a lot of data available for protein, RNA and DNA sequences. So for example, in the protein sequence example, so Uniprot is a popular database that contains a very large number of protein sequences. So as of February 2025, it had 252 million protein sequences in the database. Similarly, in the case of genome sequences, we have many databases, one of which is NCBI RefSeq database. So in RefSeq, for example, contains genome sequences of 164,000 organisms as of March 2025. And these organisms would basically cover all species from all orders. For example, they would also be large number of bacterial genomes, viral genomes, plant genomes, animal genomes and so on. So given this vast amount of data, one can ask, okay, can we train language models on for protein sequences or DNA sequences as well? And there have been many research articles that have shown that training on these sequences can also help in downstream applications. One caveat here is that observe that the nature of the data is very different. For example, if you are working with proteins, RNAs or whole genome sequences, the nature of data is very different. For example, if you just look at the average length of sequences, the average length of protein sequences is only ranging from 300 to 500 base amino acids. Whereas if you think of a complete genome sequence, that can vary depending on whether we are looking at virus, bacteria, animals, humans and so on. And in the case of human genome, we know that the length of the entire genome sequence is 3 billion. And you can easily see that there is a significant difference between the length of the sequence. So as a result, like another important factor to consider here is that the protein sequence is a very short and each amino acid is playing a functional role in the protein sequence, whereas in the human genome only about 1% of the genome is actually coding for protein sequences. A significant fraction of human genome is not functional. So because of these differences, the architectures and the learning objectives and all these parameters change depending on whether a language model is being trained for proteins versus humans genomes versus some other RNA sequences and so on. So one has to factor these domain specific information into account while designing this model and while interpreting these models as well. Okay. So in this lecture we will specifically focus more on DNA language models, and in the context of DNA language models we can again go for either masked token prediction training, or we can train the model using next token prediction objective. In both cases the model will be trained to predict the hidden character, and the model would assign different probabilities to all the characters that may be present at the hidden position. So for example, it would be the model would produce different probability values for A versus C versus G versus T, and these probabilities would vary for each position of the genome sequence. Now as part of the training process, as we know in the neural network architectures, these models would also learn the position specific context information, that is also called as embeddings in deep learning literature, where embedding is a high dimensional vector consisting of real values. So these embeddings basically can be used later, for example, to understand the semantic similarity and relationships between different positions of the genome sequence. Now, given this training process now, so once the training is done, there are many potential applications of a DNA language model. For example, functional constraint prediction, sequence design, gene annotation, gene expression prediction, and so on. So, for example, I can, based on the probability assigned by the model at each position, I can consider the likelihood of each variant, and I can do a functional constraint prediction at every position of the genome. That is, which position of the genome is functionally constrained versus which positions are not functionally constrained. Maybe I'll show you a more specific example for this in the next slide. So, a model, let's just say that a model is trained on genomes of healthy individuals, then it is expected to assign lower probability to harmful mutations in the genome, right. So, that is the general expectation that we would have from the model. Now, let us consider this example of a language model that was proposed in 2023, and this model was trained on the genome sequences of plant Arabidopsis thaliana and also the genomes from 7 related species. So, these are the genomes that the model was trained on. So, Arabidopsis is a model plant organism, and then other 7 genomes are coming from the related species that are related to this Arabidopsis species, right. So, now let us see what kind of probabilities does this model learn for different positions of the genome. So, shown here is called a logo predicted by the GPN model. So, in this logo, basically shown here, the height of each letter is proportional to its probability. What we see here is that in the regions that are functionally important, for example, these are splice donor sites in the genome, and here we can see that the model has assigned high probability to a specific character of the DNA. So, for example, at this position of the genome, the model is saying that the sequence is very likely to be GGT, the other possibilities are very less likely to occur. And actually this matches with the known functional information of this genome, that is, these regions are known to be functionally conserved between different on different Arabidopsis plant genomes. As a result, this conservation pattern is well captured by the model, and by well conserved I mean that any mutations in this genome would affect the RNA splicing as well. So, this we know from the literature, and if suppose there is any other base besides T at this location, then that may disrupt the RNA splicing, leading to alternate protein sequences. So, as a result, some of these functionally important regions like the boundaries of exons are functionally important for organism, and at these locations the model will give very high likelihood to specific characters compared to other possibilities. So, at this location what the model is saying that the probability of T is much greater than all other possibilities. So, similarly we see that behavior at splice acceptor site as well, where the model is specifically predicting that character AG is much more likely to occur compared to all other possibilities. And observe that these probabilities have been learnt in an unsupervised manner. So, there was no data given to the model about what regions of the genome are splice sites and which regions are intergenic and so on. So this pattern of learning just from genome of Arabidopsis and 7 other related species is interesting. Now in the context of human genomes, as we know, a typical human genome would have 5 million mutations on average, and in clinical settings, predicting variant pathogenicity, that is predicting which variants are likely to cause diseases, is very important in rare disease diagnosis. So in such applications, DNA language models can help in genome-wide variant effect prediction. That is to understand which variants are likely to cause diseases versus which variants may be benign. So this is a promising application of DNA language models when applied to human genomes. We also can understand, like, because these language models can have large number of parameters, it is difficult to interpret the model just by looking at the model weights. So often people use different empirical ways to understand whether a model has learned something useful or whether it has been trained well. So shown here are the embeddings learnt by the model in different genomic windows. So for example, if we look at the sequence, so what embeddings is the model learning for a region of the genome that is between the genes, that is intergenic region, versus location of the genome which corresponds to coding sequence. Similarly, introns, 3' UTR, 5' UTR region, non-coding RNA, as well as repeats. What we see is that if we visualize these embeddings, so embeddings are high dimensional vectors, but for visualization purposes we can reduce the dimension using UMAP and plot them on a 2D scale. So here we see an interesting pattern that the embeddings that learned by the model in an unsupervised manner form different clusters depending on where the genomic window is. So for example, intergenic regions form a different cluster compared to CDS, compared to intron, and so on. So what this illustrates is that the model has learned useful representations for different classes of genomic windows in an unsupervised manner, and these embeddings may be useful for further downstream applications as well. So for example, using transfer learning techniques, I can use these pre-trained models and further fine tune them for various downstream applications. An example is, let us say I want to annotate genes and cis-regulatory elements of the genome, and as an example this was done in a 2024 paper by further fine tuning a DNA language model. So as a result, these embeddings can be useful, and through transfer learning technique we can address a large number of applications where some supervised training data is also available. So we can start from these pre-trained models as a first step and further fine tune them depending on what application you want to focus on. Now if you look at the development cycle of how these models are developed, typically starting from the initial conception of idea all the way to the deployment. Then as a first step, and one of the important steps, is what data set would you want to use for training the model. So in the context of DNA language models, for example, what genome sequences do you want to consider, from which species, and so on. Like, do you want to downsample the repetitive sequences? Similarly, what architecture should we use for training? Like, there is a large choice of deep learning architectures that are available in the literature. What training objective should be used? So for example, I may start with a pre-trained model, but then it should also offer the capability of fine tuning further for different tasks. And then finally we could interpret these models as well, like for example looking at the probability assignments or visualizing the embeddings. One would want to understand whether the model has been trained well or not. And before deployment, of course, we would want to benchmark them properly, where we may want to review whether there are existing benchmarks for that type of problem, or whether is there a need to create new benchmark for that use case. So this is a illustration of the development cycle, starting from the choice of data sets, architecture, learning objective, and interpretation, and finally evaluation. So there are a number of challenges which are still open in this context, in the context of DNA language models. So for example, some benchmark studies have found that genomic language models did not outperform non-GLM baselines in some applications. That is, the conventional techniques still do better than language models in various applications, and maybe here further work is needed to show that these language models can offer significant value in human genetics. So at the bottom I have listed two references which have made these conclusions. Another challenge is it's often unclear how much increasing unlabeled data and computation will keep improving the model performance. For example, in a recent study, the authors found that a protein language model could be scaled further to only improve protein structure prediction, but other tasks such as function or property prediction did not improve if more data and computation was added. Now another challenge, especially in the context of DNA language models, is that there is a different range of scales. For example, one may be interested in looking at how the model is modeling the short motifs versus longer genes versus entire whole genomes. So far there exist models such as MegaDNA and EVO in the literature that take an entire genome as context, but they have been shown to work only on smaller microbial genomes where the genome length is only few millions. How to extend these models to complete human genome where the length of the genome is in billions is an open question. And the length of the sequence matters because there may be long range interactions between different positions. For example, there may be enhancers and promoters that are 1 million bases apart in DNA, but they may still be interacting because of the 3D form of the DNA. So because the DNA can fold inside so DNA exists in a folded format inside a cell, and that allows for long range interaction between different bases. As a result, considering long range interactions in these training models is a big challenge, which frequently makes it challenging to scale these models to very long genome sequences. So there are a number of open questions that the review article listed. For example, how can we model different scales from motifs to genes to whole genomes, right? Similarly, so far we are only looking at substitution mutations, like what is a chance of mutation A to C, C to A, and so on. But the question of how do we incorporate structural changes such as large insertion, large duplication into these language models, that is open question. Similarly, another open question is how do we integrate transcriptomic epigenetic data that is basically integrating data of multiple modalities. And also it's unclear, and can we understand better what makes some genomes harder to model compared to others. So for example, as I showed the results in Arabidopsis plant, that has been found to be easier to model compared to human genomes. So it's unclear what makes some genomes easier to model compared to other genomes. So to summarize, in this lecture we focused on genomic language models. These are the language models trained on DNA sequences. For further reading, I encourage you to look at this review article, which goes into details of what we just discussed today. And with that, we will close this lecture. Thank you.